Our little friend here seems to think bacteria are gross and useless, so we need to teach him their importance in making him grow big just like us.
Now this is a story all about how my life got turned upside down, and I'd like to take a minute, just sit right there. I'll tell you all about nitrogen fixation and all its affairs.
He seems to be yelling at the bacteria to leave, but it looks like the atmospheric nitrogen is leaving him too, which will stunt his growth due to no more protein synthesis.
Since nitrogen is a diatomic molecule, it has a stable triple covalent bond. Therefore, it must be broken down by diazotrophic microorganisms, which are only active when little nitrogen is present. Non-phototrophic diazotrophs form symbiotic relationships with plant roots, allowing the host plant to receive nitrogen and bacteria to receive organic compounds. Who can carry out nitrogen fixation, you say? Only bacteria and archaea.
Our friend seems interested in our lesson, so let's tell him how he can get his nitrogen.
Actual process of nitrogen fixation uses an enzyme called nitrogenase, which is made up of 2 subunits and 1 cofactor. The first main subunit is dinitrogenase, which uses an electron transport chain of redox reactions to pump electrons from donor proteins into the main portion of the enzyme to meet up with 2 protons to form H2, and the cofactor facilitates this process. The newly formed H2s are then used in the second major subunit called dinitrogenase to break the triple bond of N2 one by one until they are fully broken, creating 2 NH3s, also known as ammonias. This process creates the 2 ammonia and 1 extra H2, along with a hydrolysis of 16 ATPs. This process works very well, except whenever a lot of oxygen is around, because that's really bad.
It seems like he likes all of his oxygen buddies. However, too much oxygen could stunt his growth. Of course, he doesn't know about this, so let's tell him. Dinitrogenase and reductase. Those are the 2 subunits of the nitrogenase enzyme which work to reduce nitrogen. FeMo-Co refers to the cofactor of dinitrogenase. In reducing nitrogen to ammonia, electrons combine with protons, form hydrogen, and go through the electron transport chain. Environments with oxygen are not ideal due to the sensitivity of the FeMo-Co cofactor near the surface of the enzyme. Microorganisms that do live in these environments and use aerobic respiration or produce oxygen in photosynthesis must be careful to protect nitrogenase. In anabaena, cells called heterocysts are formed when nitrogen is limited, and they provide nitrogen to neighboring cells. Gene expression comes into play with heterocysts because in this new routine, nitrogenase production genes are highly expressed and photosynthetic activity genes are turned off. Look at that. Seems like he's coming around. And, oh wow, he understands the importance of not having so much oxygen with him. Our little friend now understands the importance of his symbiotic relationship with his bacteria, and look at him grow into the larger, healthy potato ready for harvesting.
Thanks for coming to my TED Talk.